I'm pleased to welcome back Dr. Turaga, who will be moderating part 3, the 2024 ACPMP Research Collaboration and Grant Awards. And just a reminder that there will be another Q&A session at the end of part 3. All right, so take it away, Dr. Turaga.
I think it's my pleasure to introduce my friend, Andriana Holovati, who's a remarkable force in the field of appendix cancer. I think she needs no introduction. She did her PhD in Detroit and then became an NIH NHGRI fellow in genomic medicine at the University of Utah. So she's had a remarkable body and breadth of experience in genetics, and she is the principal investigator of the GAP study that many of you may have participated in. And if you haven't, you should, and look it up. So she'll certainly talk about it. But she's also the chair of the Scientific Advisory Board. And so without much ado, Andriana.
Thank you, Kiran, so much. What a pleasure to be here today and to try and kind of make a miracle happen and talk about 10,000 plus words in a short 20 minutes. So I really appreciate the opportunity to talk today, partly in my role as chair of the Scientific Advisory Board for the ACPMP Research Foundation. I'm really having the opportunity to give a first preview and look into a scientific brain trust or think tank that we held, and kind of the what's coming out of that and what that means for the entire research field in appendix cancer. And so briefly, just my disclosures. So at the end of last year, first I want to preface this by saying in my role as chair of the Scientific Advisory Board, I have the privilege in working really closely with Deborah Shelton of the ACPMP Research Foundation, who is my partner in crime in this entire effort. And I have the privilege to speak on behalf of us both, as she really co-chaired this event and helped it come together from idea to action. And so we decided as part of the Scientific Advisory Board's agenda that we really needed to be forward thinking about what the research priorities are and how do we drive the field forward for appendix cancer research. You've heard so many critical topics already this morning, from KRAS therapies to early detection, potential risk reduction, et cetera. Unique needs of patients after an appendix cancer diagnosis. But how do we take all those ideas together collectively and really chart a path forward united in this field? And so Deb and I sought to bring together a curated small group of leading experts across diverse appendix cancer research disciplines to inform our research portfolio for the Catalyst Grants Program and also the field. And we sought to do this by identifying the biggest needs and gaps and really charting a roadmap for the landscape of appendix cancer over the next decade and beyond. And so we were fortunate to invite a curated group of experts across multiple disciplines, and that most individuals accepted our invitation or were able to come down to Nashville at the end of this last year. This group of experts comprises surgical oncologists, clinical researchers, basic science researchers, medical oncologists, expert pathologists, et cetera, members from the Scientific Advisory and Medical Advisory Board that span almost a dozen different institutions. So I have the privilege of presenting what came out of this on behalf of all these fantastic colleagues. Okay, so we started off this think tank. I want to kind of prime you what this event looked like, and then really spend most of my time talking about what'll be coming forward from this and giving you the first look that nobody's ever seen of this quite yet. So we decided to ask one main question. We asked, what are the most important priorities for the entire field of appendix cancer research? We asked people to reflect on this individually, and then we utilized what's called the nominal group technique, developed and explained by the CDC in some papers, to really come to a consensus or a collective agreement on what are these priorities for the field of appendix cancer research. So this was done in a multi-step process where everybody who attended the think tank shared a single idea at a time, and we kept going round and round until nobody could come up with anything new for quite a while. We then as a group put together similar ideas and were in agreement with how we did that, and generated a pretty long 2-page list of concepts. And then we asked everybody in the room to vote. to rank from most to least important their top 5 priorities for the field of appendix cancer research, actually looking at the card that we gave participants. We then tallied all those votes and really came to a main consensus. We actually had a tie, so you see 6 different columns here. But what I'm going to spend my time talking to you about today is what are these research priority areas? I'm going to give you a brief flavor into some of these and really encourage where you can learn more over time. These are in no particular order, I want to advise. This is kind of just how the story flowed together overall. And so when we define these 6 research priorities, we actually did some deep dives and small group discussions of diverse experts and had everybody at the think tank kind of percolating and diving into each of these priority areas. Then we came together as a group and discussed them in detail. So you're going to see some of those key spark notes or key points from each of these areas as I talk through them briefly overall. The first of which may come as no surprise, and it has to do with appendix tumor pathology. So our first priority area really was refining appendix tumor pathology and histology and our histopathologic classifications. Those are some big words, so what does that mean? I think the tumor board this morning gave a great insight into what it looks like to see appendix tumor cells under the microscope. And this is crucial because our understanding and how we treat appendix tumors hinges upon an accurate classification of this disease using that gold standard technique. And so one of the first main areas that came out of this priority area was the continuum of histological features. Appendix tumors are not just one histology. We've heard in the chat people ask about goblet cell, signet ring cell, talked about lamin in the tumor board, etc. So actually, and what will be forthcoming from this work, you'll see a snippet of some of the characteristics put together by our pathologists, including my dear colleague Kay Washington, who chairs the entire AJCC editorial board for clinical staging of the 9th edition, in different snippets of what pathologists look for, what are some of the features that define each of these histological subtypes that you all have been talking about. You'll also see representative images forthcoming for each of those and what those tumor cells look like under the microscope. We also talked about tumor grading and the different grading systems. AJCC has one, the World Health Organization also has one, and sometimes there's a backwards approach using peritoneal tissue to work backwards to define the primary tumor. And so when you look at peritoneal mucinous tumor cells, how those are defined across different schema actually differ. And so we talked about the importance of understanding this and moving forward in this space for accurate classifications. We also really focused on digitizing histology for appendix tumor detection. What does that mean? That means scanning in that slide that you saw on the tumor board today and allowing for an expert pathologist to be able to read it, even if they're not at your institution. So I think Dr. Turaga's peritoneal surface malignancy consortium guidelines really speak to the importance of having an expert pathologist review the tumor tissue under a microscope. And this digitization of histology really helps us break down geographical or resource barriers towards that. And we also spoke about computational approaches. Can we use AI or computational pathology to help us determine tumor grade or what that cellularity looks like, predicting tumor subtypes, et cetera, moving forward? So for the sake of time, I'm going to breeze through these. Happy to take any questions. I'll tell you where you can learn more. The second priority area was the molecular characterization of tumors and targets, which you've heard a lot about today already. This is essential to understanding the pathophysiology of appendix tumors, but also for new target and new therapeutic discoveries. What does that look like? The integration and standardization of molecular profiling in a patient care, that every patient should get a tumor spell check to understand what changes in the tumor DNA there are that might be able to be exploited. Also potential for genetic testing. Heard a lot about KRAS. The green highlights spelling changes perhaps in the DNA of mucinous appendix tumors that offer opportunities to possibly exploit this. There's also an importance to understanding the normal appendix epithelium, to establishing a composite multi-layer view. The human body is not in 1D. How do we add multiple different layers of proteins, of transcripts or genes, and of the DNA to better understand appendiceal tumors? the advantages to tumor biobanking, and ultimately addressing appendix tumor disparities. So we talk about standardizing molecular profiling. We want to make sure we're not exacerbating the disparities in who has access to that sequencing technology and opportunity. Moving forward, and I'm going to touch only upon this briefly because I know my colleague Dr. Wagner is going to be speaking a bit more about the tumor microenvironment in appendix cancer. This ecosystem surrounding the tumor plays a key role in tumor progression and how tumors respond to treatments. So an important priority is disentangling what that looks like around the primary appendix tumor, but it also is understanding the biological mechanisms of metastasis. So how do we develop a peritoneal molecular atlas for tumors that originate in the appendix? And how does the peritoneal microenvironment influence the progression of the tumor? This is briefly illustrated here. For the sake of time, I'm not going to walk through this in depth, but I think you can appreciate the number of different molecules and different colored items on this page and how many players there really are that may be, contributing to that ecosystem or microenvironment, including the microbiome and dysbiosis and what Dr. Turaga was just speaking about, particularly with Fuso. The next priority moves on to disease-specific models and a model repository. So models are really important to us in the research setting because these are key tools to help us investigate tumor biology and support preclinical studies of therapeutic strategies. So growing cells in 2D and 3D is really challenging because of the difficulties in collecting fresh tissues from primary appendix tumors, but it is an important step in understanding how that tumor grows and develops. In addition, preclinical animal models will help us test therapies before we move them into clinical trials. And this kind of points to the importance of establishing a repository or really a centralized bank or resource to support this research. We actually queried an inventory of existing appendix cancer models, and I can count them on two hands in the number of papers that have been published thus far. And one of the things you might appreciate from this brief figure is these are all mucinous tumors. And we don't have any information even on patient race and ethnicity on any of these models to ensure diversity in the work that we're doing. So while this is a great start, there's really a place where we can move forward with much more of this work. Priority 5, prospective investigations in clinical trials. This research is crucial to inform evidence-based medicine and further understanding. Prospective clinical cohorts and studies. How do we partner with you, the patients, to overcome real-world challenges? I think it's been said, and I so appreciate it. The GAP study is one of those opportunities. We're just about approaching our 2-year anniversary and are working our way towards 600 patients on GAP already. And we hope to increase, if not double that even further. This not only helps us understand about a potential genetic link to appendix cancer, but also work with colleagues across other spaces. We're reviewing pathology and collecting tumor tissues to better understand aspects of the biology to address some of these gaps. In addition, interventional clinical trials, I think Dr. Shen's group is doing great work in this space with some of the work they've already done and are moving forward in to provide clinical trials and opportunities for new drugs in the appendix cancer space. Novel blood-based biomarkers for disease surveillance and outcomes, Dr. Turaga's group, Dr. Shergill's group, really doing additional fantastic work to help chart that path forward. You heard the challenges about imaging in the appendiceal cancer space. That's an area we've identified that we have the opportunity to chart a path forward for, and ensuring the quality of and equity in healthcare delivery and outcomes. The last priority, can kind of tell we've really shifted from cells to society here, is understanding appendix cancer on a population level that really can help us deliver optimal cancer care to patients. This looks at elucidating exposures, risk factors, and patterns. You heard Dr. Turaga talk about it, and my lab was the first to show 1 in every 3 appendix cancer patients is diagnosed before the age of 50. That a disproportionate early onset disease burden is something we really need to address as these patterns continue to increase and more patients are diagnosed. evidence-based survivorship care plan. What are the needs of this growing patient population experiencing a unique cancer journey? We posit that across different domains, from the educational to the practical to the physical, psychosocial, as well as health promotion and prevention space along the appendix cancer care journey to ultimately improve patient outcomes. That also looks like ascertaining real-world patient data with multi-institutional databases, utilizing some existing cancer registries to the best of our ability to understand disease trends, and of course, provider and public education and resources in terms of keeping occult appendiceal tumors in the differential diagnosis for patients, especially younger ones who present in that way. All in all, we're really fortunate that we have put together a 10,000-word expert recommendation article that goes into every single one of these details and way more, that is expected to be coming out in the next couple weeks in defining the first ever Cells to Society research framework for appendix cancers. Like I said, it's 10,000 words. It's meant to be for a broad audience with no familiarity in appendix cancer or limited familiarity to those who are experts in the space. And I cannot thank Deb enough for believing in this idea and helping me champion it, as well as to the ACPMP Research Foundation for really bringing this together. We hope our Catalyst Research Grant Program will envelop these 6 research priorities moving forward. And all that said, this wasn't possible without Deb and the ACPMP Research Foundation, without all the fantastic contributors who took their time to come to Nashville to share their intellectual ideas, to the Vanderbilt Ingram Cancer Center for hosting and supporting that, to our fantastic patients, not just the 3 that are listed on the screen who helped contribute to patient video or that survivorship framework that you saw, but every single one of you on the call today. I would be remiss if I didn't shout out to the amazing patients on the GAP study, many of whom I'm privileged to personally know, and honored to have you a part of that work. To my lab, who really helped contribute to this event running smoothly and does the day in and day out work. You can see my team here at the virtual ACPMP walk from this past summer that we were fortunate to do. On a Nashville Greenway, and also to others who provided their technical assistance. My information, as well as that for GAP, is on the bottom of the screen, and I think hopefully I'm right about on time. I apologize for having to speak fast and covering so much information, but the future of appendix cancer research looks bright. We feel that these priorities that span from cells to society have an opportunity to chart a path forward for all of our patients and really help guide researchers, those who are experts in the space and those who may just be starting out in the appendix cancer research space, to really change patients' lives moving forward. And thank you so much for your time and for listening.
Well, thanks a lot, Andriana. That was wonderful. And I think we'll have some time for questions at the end. Our next speaker is my good friend, JP Shen. I think you all know and heard from him already this morning at Tumor Board, but JP is an assistant professor in the Department of GI Medical Oncology at MD Anderson. And his research has been to understand the cancer genome, and you'll hear some remarkable work from him. I think he's also been leveraging genetic relationships to repurpose approved drugs from the FDA, and has numerous projects in the space of appendix cancer. His background training did involve a lot of chemical biology, functional genomics, and clearly comes from his undergrad time at MIT and the Broad Institute. So an incredible superstar that I think we're lucky to have thinking about appendix cancer. So JP.
Awesome. Kiran, thank you for the very kind introduction. I'm really excited to be here. And really, thanks to the ACPMP for funding our research, and everyone who has donated to the ACPMP to make the funding possible. It's great to hear that the grant programs are being expanded. Let's see. Okay, let me advance the slides here. I have a bad tendency of always trying to pack too much in to each talk. But I just wanted to, it's been said multiple times, but it really is critical to think that colon cancer is not the same as appendiceal cancer. Dr. Turaga mentioned that we see patients that come in a lot, having been treated with colon cancer chemotherapy, which is fortunately probably not helping them very much. And it's really unfortunate. You know, a big part of that, actually, there should be like a big X through this part of the NCCN guidelines, which are kind of the Bible for most medical oncologists. You know, for basically from the beginning of the guidelines, you know, in the 70s until now, the recommendation has always been to use colon cancer chemotherapy, which is unfortunately why, you know, most community oncologists do that. There are going to ultimately be updates to this NCCN guidelines, but it's very slow in coming. And so just wanted to highlight the work that Dr. Turaga with his team at Yale and many others who've been on the call today and others have put together, that there actually are now consensus guidelines that are very soon to be in press that you know, I think have really just a much better outline of how to manage these very diverse diseases. The you know, the number one problem in appendix cancer is a rare tumor, but it's not that much rarer than other tumors that have a lot of FDA approved treatments like cholangiocarcinoma, gallbladder adenocarcinoma. A lot of subtypes of leukemia are less common than appendiceal cancer. But I think one of the big problems, other than this you know, kind of outdated idea that appendiceal tumors are just a subset of colon cancer tumors, is that there's just so much diversity from one type of appendix cancer to the next. And we've talked about how critical the grade is, you know, that high grade tumors are really a different disease from low grade tumors. But the other major factor is the histopathologic subtype. And it's great that we talked about it a little bit earlier. You know, that the goblet cell tumors are very different than mucinous tumors of the appendix. Goblet cell tumors make up maybe 10 to 15% of appendix cancer. If there's maybe 5,000 diagnoses in the entire country every year, that means there's only about 500 or so goblet cell tumors in the entire country. And so being that rare, it's been hard to study them. There's very little that has been published on these. And you know, if you're a patient with goblet cell cancer, it's very frustrating because you know, very few people, you know, physicians really don't know much about your tumor. It's also been very confusing in that over the years, these tumors have been called by many different names. You can see many of them there. And so if you're looking at, you know, older literature, which is, you know, even from like 10 years ago, like you'll see the term goblet cell carcinoid, which is not really preferred anymore. It's better to call it goblet cell adenocarcinoma, but that's kind of added to the confusion. And these are really, fundamentally, these are really kind of complex tumors in that within one tumor, you have cells that are different. You have both the traditional kind of epithelial cells that we would think of in an adenocarcinoma, but you also have these neuroendocrine cells, so cells that kind of have hormonal features that would secrete hormones and mucin into the normal GI tract that have become cancerous. So it's complex. It's a complex and heterogeneous mix. You know, and then sorry, I'm struggling with the slide advance here. On top of that, you know, because you know, in general, we have very little high quality data to know how to pick what is the best treatment for our patients with appendiceal cancer. You can see there that there's only been 8 total prospective studies ever done, you know, in appendiceal cancer, and you know, none of them specifically in goblet cell. And this was as of about a year and a half ago. Really nice review from Dr. Mohn in Australia. And so because we have, you know, we're very much trying to do more clinical trials. We have a lot of industry, you know, pharmaceutical industry is starting to get excited about this. If you are on the call and happen to be a pharmaceutical company executive, please message me and we will talk because we definitely have the patience. But in the meantime, you know, clinical trials are very slow. You know, from the time you have an idea about a trial to when it actually executes is measured in years, not weeks. And our patients need answers now. And so we're trying to supplement that as much as we can with what's called real world evidence, which is basically aggregating all the outcomes of the patients that we do treat and learning from that. And so, you know, one of the biggest strengths we have at MD Anderson is our volume. I believe we're the largest academic cancer center in the country. Across the entire GI department, we see, you know, more than 3,000 GI cancer patients every year. And for you know, but even at MD Anderson, you know, 70% of the patients that we see aren't going to go on a clinical trial. And for a long time, we weren't really learning that much from them. And so we partnered with the company Palantir to build this web-based kind of graphical interface, so we can aggregate all of the information that's already contained in our electronic health record, aggregate that, and hopefully we can learn, from these thousands of patients that we're seeing. Also want to highlight this is primarily the work of two very talented brothers who are foreign medical graduates from Egypt who have served as postdocs in the lab. And so this is by not any means complete. The way that the information goes into the health record is very disjointed. It's not always structured. Like as a patient, you might see us at MD Anderson, but you're also going to see Dr. Turaga at Yale, and you're going to see your local oncologist, and those records don't always get integrated. We're working on kind of ways that we can overcome these problems. So by no means complete, but like the Death Star, it's fully operational, and it's taken a lot of infrastructure to get to this point. But we're now, I think, starting to really see the rewards in terms of what we can learn. And so, we're still at the very beginning of understanding goblet cell cancer, but we've built out this database, and we're just starting to explore it. So we've got nearly 4,000 total patients with appendiceal cancer, and we included in there both Hammond and Lamond to get to that number. Subsetting to the patients with goblet cell, and I apologize, this is literally hot off the press from last night, which is why the figures don't look very fancy and it's kind of hard to read. But we see about an average of 35 goblet cell cancer patients every year. This has been reported before, but there is more than expected female predominance, which is interesting. And as has been commented before, almost all the time it's a high-grade tumor. So we because this is getting scraped from our health record, it actually says 13% of the time we can't actually figure out what the grade is automatically, and so we have to kind of go back in and chart that manually. So probably this is more like 75% of these tumors are grade 3 high grade, which fits with what most people have experienced. And so we are, digging more into the demographics and mixing that with the outcomes. And so we can see that one of the first things we found is that the early stage, so stage 2 or stage 3 patients seem to do very well, but there's huge variability in the stage 4 patients. And so, staging, right now, the staging of goblet cell cancer has basically just been copied from colon cancer, and it doesn't really pick up the nuances that help us decide, what is going to be the best treatment for each patient. And so we're working on trying to use, tumor markers, like CEA, CA19-9, CA125, specific mutations, potentially the Ki-67 score, which is actually very helpful in neuroendocrine tumors, but I think might be helpful in appendiceal cancer, to try to help us better identify who are the highest risk patients, and who are the patients that are lower risk and maybe don't even need treatment. And so this is unpublished data, but we presented this as an abstract, but hopefully the manuscript will be ready for submission soon. One thing we found is that even though the national guidelines suggest that stage 2 and stage 3 appendiceal cancer patients be considered for chemotherapy as if they were colon cancer, that there's really no high quality data that suggests that the patients benefit from that. And so looking retrospectively, if you restrict it to just the patients who had surgery at MD Anderson, because we know everyone we operated on, we know who relapsed and who didn't relapse, only 7% of the stage 2 or 3 patients relapsed, and the numbers were particularly low for goblet cell. So interestingly, the risk of relapse for a mucinous tumor was significantly higher than goblet cell. And you can see here these curves that basically, if you got adjuvant chemotherapy or didn't get adjuvant chemotherapy, basically your survival is excellent either way, suggesting that you really don't need to do the adjuvant chemotherapy except in some particularly high risk situations. Again, retrospective data, but it suggests that I don't think there's ever going to be a prospective study of adjuvant chemotherapy in appendiceal cancer, and so it suggests that the benefit is marginal. One other thing we looked at is that what are the risk factors for whose tumor comes back? It differs. One, they differ from colon cancer. That's first important thing to remember. But the risk factors for mucinous appendiceal cancer are quite different than the risk factors for goblet cell. For example, virtually every goblet cell tumor has perineural invasion. That's because these are kind of a neuroendocrine type mixed hybrid tumor. And I've seen a lot of people that, community oncologists will say, oh, well, perineural invasion is a risk factor for relapse in stage 2 colon cancer. So that's a reason to give chemotherapy to a stage 2 goblet cell patient. But in reality, every stage 2 goblet patient has perineural invasion and most of them don't relapse. And so I think it just really speaks to this idea that we can't just assume that appendiceal cancer is going to behave like a more common cancer. We really have to study appendiceal cancer itself, and we have to study the different histologic subtypes independently because they're very different. And so this is just kind of showing you this in Kaplan-Meier plot that basically I think we had maybe 30 grade 1 or grade 2 goblet cells. Not a single one of those patients relapsed. Lymph node involvement was a significant risk factor for relapse, but even in the, so the relapse rate was very, very low in the lymph node negative. So stage 2 goblet cell had a very, very low relapse rate, but it was higher in stage 3. But even in the stage 3 patients, you can see here that better than 75% of them are still alive at 5 years. And so interestingly, and this actually kind of relates to some of the questions about follow-up, when the goblet cell tumors do come back, it actually can be quite delayed. And so it's probably too small to read on your screen, just like this on mine, but I think that says median 31 months or maybe 38 months to relapse in goblet cell versus only 11 months for colonic type and about 22 months for mucinous. So the colonic type when they do relapse, seem to be fastest, followed by mucinous, and goblet cell can actually have a very delayed, the overall risk of it coming back is low. But it can happen quite late, suggesting that we really do need to follow these patients for extended period of time. Okay, and so that's kind of what we've learned from the clinical side, aggregating the clinical data. Where I was very tempted to put some of our preliminary data about the chemotherapy findings, but it's really very preliminary. And if I'm invited back to give a talk next year, we'll definitely have more information on that. That's definitely the thing that we as a medical oncologist we care about the most is what is the best chemotherapy for this patient, and we think that we're going to be able to learn from this, but the way that this information is reported, the way we type this into our notes is not honestly, we're kind of the worst in terms of being disorganized in terms of aggregating this, and so it's harder for us we're using now machine learning process and natural language processing to try to build this out, but it's a little bit in progress. But based on what I just showed you, that the clinical outcomes of these goblet cell patients are very different than the mucinous tumor is very different than colon cancer. It's maybe not surprising that when you look at what genes are mutated in goblet cell cancer, it's very different than what we see in mucinous adenocarcinoma on the far right, or non-mucinous or enteric type in the middle, or signet ring cancer. And you can see here that this is our MD Anderson sequencing data from just most of these patients had about 140 genes sequenced, the 140 most commonly mutated genes in colon cancer. Now most panels are more like 300 or 400 genes. But if you look at the most frequently mutated genes in cancer, very few of them, you don't see very many mutations in goblet cell tumor. The most commonly mutated gene is P53, but that's only like 23% of patients. And that's, the number one most common mutated gene, suggesting that we need to look outside of these very commonly mutated genes to see what is really causing goblet cell cancers. Obviously, all cancers have to be driven at some stage by mutation. And so, sorry to go through this kind of quickly, but we have a very exciting collaboration with the company Moderator, who by nature of a tissue-informed ctDNA assay, basically the tumor and the patient's germline are both sequenced, so they can identify what changed into the tumor and therefore track it in the blood. But all of the exome data, the sequencing data for these tumors is aggregated, and they've been kind enough to allow us to research in collaboration with them. And you can see here that the overall mutational burden is pretty low, only 2.8 mutations per million bases, which is low. But the genes that are mutated are actually not ones that we have seen before. You have, again, it's kind of small, I think, but P53 is a well-known cancer gene. SMAD4, also a well-known cancer gene. But all the other genes on that list are not very well known. You can see KRAS is way down the list. We're very excited about that first gene, which is a zinc finger protein. I think it's bigger on the next slide. Yeah, this gene ZFP36L2 is a zinc finger protein, which basically means it binds to the RNA after the RNA has been translated and helps stabilize it. And so we think that when this gene is not functioning, we see big changes in what genes are turned on and turned off. And we think that the second gene, WTAP, which is Wilms tumor associated protein, also kind of works in a similar fashion. And we think that this is maybe a theme in goblet cell cancer, in that the reason that you can have these relatively aggressive tumors without many mutations is that the mutations that they do have aren't changing just one gene, they're actually changing the expression of lots of genes. And so in that way, you don't need lots of mutations to get, the cancer phenotype from these goblet cell tumors. And so we are in the lab, we are in the process of knocking those genes out to kind of replicate what we see in the tumors to validate that those are in fact cancer mutations. And so this is maybe a little bit esoteric for the non-scientists, but I just wanted to kind of put this in there. And so, one of the ways that we can try to deal, the big problem in cancer is the heterogeneity. We know that, each tumor is like a snowflake, unique at some level, but we can't have, one drug for every patient. And so how do we know how, what patients should be grouped together, what tumors are similar enough that they should get the same drug? And so if we, instead of treating each gene as something independent, we know a lot about how the genes interact with each other, how they form networks. Like, these genes make a protein product that all these proteins bind together to form a complex, and this complex acts on another complex. And based on, years, decades of work throughout the scientific community, we now actually have very well curated networks of how cancer genes work together. And if we take the mutation data from the whole exome, all 20,000 genes that have been sequenced, we overlay it onto these networks, we can now cluster the tumors into similar groups. And now doing this, we're starting to make some interesting discoveries, and we're starting to kind of actually see some structure, because it's very difficult to, if you just have 20,000 genes of which you might recognize the names of 1 or 200, what do you do with all this information? Now that we can use the networks, we can cluster the patients, and we're starting to actually see that the clusters line up with some of the well-known driver genes that we're aware of. So the tumors that have mutation in that ZFP protein all seem to cluster together here in the middle. There's not too many KRAS mutant goblet cell tumors, but they are clustering together with, interestingly, with SMAD4. The P53 mutant tumors form a separate cluster. This other gene, MLL-T4, which is a post-translational modification gene, like an epigenetic regulating gene, also seems to form a unique cluster. And so we also the Rho-A, which is Rho kinase associated protein, which is thought to activate the RAS-MAP kinase pathway. You can see that those tumors are forming a unique cluster here. So, what does this exactly mean? Short answer is I don't know yet, but we are now starting to, we think that once we group the tumors into these groups, we think that the drug response will be different, depending on the molecular class. And so that's really the next step. We were pairing this with the ability to look at who got what drug and who responded, to see, can we tease out, can we start to use this information to really make predictions that people care about, like, this is the, of the 3 drugs I could choose from for you, this is the best one for this patient given the specific mutation pattern. And so so we're really hopeful. So that's DNA sequencing. The next area that I'll talk about just very briefly is that we're now starting to do RNA sequencing. This was work that we just published a few months ago, where we did just 4 patients, where we did single cell sequencing. In this case, we take the tumor, we just digest it a little bit. So every cell in the tumor, there's tens of thousands of cells in the tumor, and we sequence each cell individually to see what genes are turned on in each individual cell. And the first thing that we learned from this is that the vast majority of the cells in these appendiceal tumors, it's not actually the tumor cells. You have a lot more either immune cells and fibroblasts or stromal cells that are kind of supporting the tumor cells than the actual tumor cells themselves. And that's part of why when you look at it under the microscope, a lot of times you see just a thin strip of tumor and a lot of reaction to that. And that has important, we think that this is part of, this is just kind of showing that when we do this, you can see that the goblet cells are very different than the mucinous tumors. And so it's not really, single cell sequencing is not really possible on every patient. We've only done 4, but we can now do bulk RNA sequencing on the vast majority of patients. And so we collaborate with primarily the company Boston Gene, although Caris also does this. We also have a research collaboration with them. And so from the RNA sequencing, you can actually identify what are the cells. You don't actually have to separate each cell individually to determine what is the cell composition. Based on the different signatures of the different cell types, you can kind of deconvolute, basically figure out what fraction of this is tumor, what fraction of it is immune cells, what fraction of it is stroma. And you can do this on, we do this, I order this test, 2 to 3 times every week on virtually every new appendix cancer patient that I see, because we think that, we don't really know what patients are going to respond to immune therapy, but it's been shown in other tumor types that if you have at baseline a lot more T cells in your tumor, the chance that you're going to respond to a T cell activating therapy is better. And so do I know that for a fact in appendix cancer? No, but given that we don't really have a lot of other high quality data, we were kind of using this as a way to prioritize the drugs that we do have. And so the other thing that we're kind of learning is, so we can look at not only what types of cells are there, like do you have a lot of T cells, do you have a lot of macrophages, do you have a lot of fibroblasts? We can also look at what pathways are getting activated, and virtually every single appendix cancer that we profiled has a very strong angiogenesis signature, which is why we think that the angiogenesis inhibitors are a big part of the systemic therapy. I didn't put this slide in, but we have some retrospective data to suggest that, for patients that get bevacizumab, they have a better survival outcome versus those that don't. Obviously there are also potential risks of that medicine, but in this way, we're starting to let the RNA sequencing kind of help direct what is the best therapy for each patient. So we're one expression, which I don't necessarily love, but it's maybe the best way to say it. We're kind of like building the plane and flying at the same time. I mean, do we have, would we like to know more about what this means? Absolutely. Do I understand it completely? Absolutely not. But I do think it's available, and I do think it is helping us to make decisions in real time, because as the other medical oncologists will tell you, it's very challenging to know what is the best treatment for each patient. And so I think that's the last slide. I know I've gone a couple minutes over, so I appreciate that, and I apologize for going through this quickly. Thanks again for this opportunity to speak, and to our collaborators.
Well, thanks, JP, and I hope all of you on the symposium, get a flavor of how exciting the research is in this space, and potentially its opportunities to unlock numerous therapeutics for this. So thanks for joining. JP, if you don't mind, we'll just keep moving along with our presentations. Our next speaker is Dr. Wagner, who is going to be talking a little bit about the immune microenvironment in appendiceal cancer in the peritoneal cavity. Dr. Wagner is the Director of Complex General Surgical Oncology at Allegheny Health Network Cancer Institute, a good friend, great surgeon, and has been doing some remarkable translational work. So, Patrick.
Thank you, Dr. Turaga. Can you hear me okay? All right, and the slides will come up, I believe. There they are. Okay, and I'm going to speak under the assumption that we have a mixed group of some clinicians taking care of patients, some patients, some advocates, and some caregivers. We've been presenting our research at a couple different forums lately, so some of you may have seen a few of these slides already, but Most of the work is new, and frankly, I think I wanted to take an opportunity today, given that we received the Catalyst grant this year, just to show what we're doing with the funds that were provided by ACPMP. So first and foremost, thank you to the foundation for the opportunity with the grant support and for the presentation today. I'll try to focus on what we're doing with that. Okay, so the next slide. Or do I have control? Let me just,
Yep, you have control.
how do I,
You can click. There's
oh,
arrow.
got it. Yes, got it. Okay. All right. So I had just a few disclosures regarding funding, one of which is the ACPMP Foundation. None of them are a conflict of interest. A lot of us talk about the tumor microenvironment these days. And for those of you who aren't aware of what that means, I would say traditionally in cancer research, there's been so much focus on the tumor cells. And as time has gone on, we've learned to focus an equal amount of attention on the background tissue, the connective tissue, and the immune tissue and other tissues that are not normally what jump to your mind when you think about a tumor, but there's a dynamic environment in which a tumor grows. And many of our emerging ideas about therapy are directed at that, including immunotherapy, which everyone has heard of, but also other kinds of therapy directed at blood vessel growth or the metabolism of tumors and other things on the horizon, even exciting things like microbiome research, where the bacteria that are present within a tumor environment can be targeted in some ways to help control cancer. For one other thing is that if we take tumor cells out of patients, which we often do, and grow them either in a petri dish or in an organoid or even in an animal model, we lose that environment or replace it with some other environment. And so it can be very hard to know sometimes how to interpret studies that are done without really giving a lot of thought to how is this going to work in the human environment. Sometimes things that work great in the lab don't work well in patients, and this is probably one big reason why. And so the big theme that you hear many of us talking about, and actually was the title of the grant proposal that I had, was to look at this microenvironment in a handful of different ways to really consider the microenvironment as an entity in appendix cancer worthy of its own investigation. So for appendix cancer, the microenvironment's different than it would be for other cancers. The appendix itself, the organ, not the tumor, but the organ in which the tumor originates is one element of it. There's the tumor itself. There's the peritoneal tissues to which appendix cancers often spread. Then in late stage cases, there's fluid or ascites that builds up around the organs, and that is an environment that needs to be considered. And many of you are probably well aware, especially from the pseudomyxoma side of things, that mucin and scar tissue can form in and among the organs in this environment. And so all of these things are really important to think about, almost more so with a cancer like appendix cancer than other cancers, although it's true in every other cancer as well. And so that is sort of breaking down the different aspects of the microenvironment, or which compartments or entities are involved for appendix cancer. I'm going to show you in a few different slides in a few different ways that the tumor microenvironment does dramatically influence appendix cancer biology. Some of that Dr. Shen was just driving at in terms of figuring out which cell types are present in the environment and how they influence outcomes or response to treatment. I'll talk a little bit about that. Also, just to know that, the tools are emerging to define this environment. And then probably what I'm most excited about, what we're doing is developing tools to try to alter that environment in ways that are helpful for cancer patients. So just very quickly, I want to emphasize how unique the appendix is as an organ. It's very different than any other part of the colon or intestines. It has immune tissue that's not present anywhere else, a type of immune tissue that's present nowhere else in the intestines. And so I think sometimes we gloss over the appendix as just a vestigial organ or an organ that or a extra part an extra part of the colon that, but it's important to know that in normal health, the appendix is important in producing antibodies that go into the intestine and to serving as a locus for the microbiome, for instance, after a diarrheal illness or after a bowel prep or some other reason why the microbiome of the colon is wiped out. The appendix is thought to serve as sort of an inoculum of bacteria that can go back into the colon and reconstitute it. So if you take antibiotics, for instance, and it wipes out all of your normal bacteria, the appendix is out of circuit in some ways, and so it can reconstitute that dynamic environment. So I'm going to talk a little bit about some of the work that we're doing now, and this will be replaced over time with higher tech methods, some of which Dr. Shen was talking about with single-cell RNA sequencing or multi-parameter staining, but just sort of to simply look at the lymphocyte or T cell counts in tumors. This has been done in most other tumors by now. In colon cancer, there is a lab test called the immunoscore, which takes two different types of T cells, or excuse me, two different markers for lymph cells, and compares them and averages them and turns that into a prognostic score for colon cancer. One question that we had is whether the same thing would be true in appendix cancer. It stands to reason. In many cancers it is, and in fact we found that lymphocyte counts correlate strongly with grade, and that making parameters out of the lymphocytes can allow for us to stratify patients into higher and lower risk groups. We also feel that some patients that have an extremely high number of lymphocytes or T cells in their tumors may be ideal candidates for immunotherapy as it develops in the future in clinical trials, or if we're looking for a subset or groups of appendix cancer patients that might be more likely to respond to immunotherapy, the outliers in this regard would be the ideal set to look at first. I'm just going to go quickly through this. Again, we sort of want to feed back to the foundation that how the money for the grant is being used and put to good effect. We're also looking at macrophage subsets here in work funded by ACPMP and comparing the amount of macrophages, which play a number of different roles in the immune system, but they also contribute to the fibrosis that occurs in tumors. And just to show early correlations with different types of appendix tumors of different grades, as well as between the peritoneal tumors and primary tumors, and correlations with the amount of cancer in terms of the PCI or the peritoneal carcinomatosis index. And here we are just comparing the peritoneal and primary tumors. And then I'll move on quickly. I know this is kind of breakneck speed, but there's not much time to cover a lot of ground. We began some studies looking at microbiome and appendix cancer. There is a species of bacteria called Fusobacterium nucleatum, which is of growing interest in colorectal cancer. It's strongly associated with some types of colon cancer and suspected, although not fully proven, to have a causative role, to be a causative agent in some cases of colorectal cancer. It's also known that the appendix harbors this bacterium, and in patients that have appendicitis, the levels of Fusobacterium are a lot higher. We wanted to study this process in tumors to see if, number one, is this bacteria present in appendix cancers? And if so, were there any correlations to draw? And so we have a paper under review at this point, looking at just sort of describing for the first time this bacteria in appendix cancers, as well as in a background of normal appendix specimens and appendicitis. And just to show some early findings from that work, we did find a correlation with the grade of tumor, as well as with the patient age, and also some lymphocyte subsets based on the amount of Fusobacterium in the specimen. This is very early data, and we're going to look at a lot more bacteria besides Fusobacterium, but this was the most obvious first look given its role in colon cancer. And so I'm excited to have this process underway. And here, this is just a Kaplan-Meier curve, which shows survival differences in patients with and without higher levels of this bacterium. And it actually survival is a little better with higher levels of fusobacterium. So a lot more to come on that. And this is a throwback slide just because I think it's important to, microbiome is a hot topic at this point, but this is not the first time it's been studied in appendix cancer or pseudomyxoma. This was an old study from 2008 by Dr. Sardi's group, in which a specific bacteria, H. pylori or Helicobacter pylori, and other bacteria were studied in the mucin of pseudomyxoma and were found there. So I think we have a lot better tools now than we did in 2008, and we know a lot more about the role of microorganisms in cancer. And so I'm excited to unfold that work over the next remaining year and a half of the grant, and including some, hopefully some very high complexity projects looking at both the circulating blood in patients with appendix cancer as well as tumors. So that's a work in progress. And now I'm going to switch gears very quickly, and this is a lot to fit into 15 minutes, but I do want to talk about some of our open clinical trials in the realm of changing the microenvironment. So we talk about the microenvironment as if it's a fixed entity, and to some degree it may be, but we've done work just really trying to drill down on exactly what's different about the microenvironment in the presence of carcinomatosis or pseudomyxoma or other cancers versus the normal state, finding different immune mediators, or we call them cytokines, but you can think of them as hormones for the immune system. They're signaling molecules that go from one cell to another, and really just studying carefully which ones are present at higher levels in cancer patients to look for targets for potential clinical trials. So without getting too far into the biological details here, just to show you that a big part of the grant is going towards characterizing this environment, looking both at the fluid in the peritoneal cavity as well as the lining, the background lining, or the soil, so to speak. If you think of peritoneal tumors as seeds that grow, what they are growing on or in is the peritoneal environment. So really studying that carefully and thinking about interesting ways to try to change that in a way that might be helpful for patients. And one early finding that we've had, and this is work directly sponsored by the ACPMP grant, was looking at proteomics or protein pathways that are different between the patients with carcinomatosis and patients that don't have carcinomatosis. And one of the things that we found that we're excited to follow up is a high level of neutrophil activity in the peritoneal cavity in carcinomatosis. Neutrophils are immune cells that are kind of triggered to respond to bacterial infections or other immediate stress responses. And probably in the context of cancer, neutrophils are very maladaptive. They don't do much to fight cancer, and they probably secrete a lot of chemicals that accelerates cancer growth. So we think there's a lot to follow up on here. We did a couple of other assays to try to chase down this result, including looking for neutrophils in the body cavity or in the mesothelial lining or the peritoneal lining, and found huge difference between patients with or without carcinomatosis. And this work will be presented hopefully at the SurgeOnc meetings in the spring. And in the meantime, just to say that tying it all together, there was evidence in the peritoneal fluid and in the peritoneal tissue, and actually looking under the microscope at the cells in the peritoneum, that neutrophils probably are contributing, which in retrospect makes sense given that neutrophils secrete a lot of chemicals that can lead to inflammation, scar tissue, and also just impair the immune system's response the way it should be, which is to react against the cancer cells. So very briefly, talking about reconditioning trials, we have a pair of clinical trials that I'd like to just go over quickly. One of them, they're called RIOT-1 and RIOT-2. RIOT is an acronym for Regional Immuno-Oncology Trials. So the first one was an experiment to inject peritoneal tumors with a stimulant of the immune system. It's actually the stimulant is actually derived from bacteria. There were no living bacterial cells in the injection, but it was a chemical component of bacteria, which is a major potent stimulant of the immune system. And so in any case, this was a 12-patient study injecting both tumors as well as background tissue with this bacterial chemical called lipopolysaccharide, or LPS. And then a control, each patient also got an injection of saline, which was to give us a comparison to really look at specifically what was being triggered by the lipopolysaccharide. A lot more work to be done on this, but a very first sort of bare bones assessment showed us that we did see measurable changes in the immune cells within these tumors. And this is just looking at 3 or 4 different markers, but we'll be looking at this with hundreds of markers over the near future, and really drilling down on what we can do to change the microenvironment, hopefully to wake up the immune system and cause a response, either as a standalone intervention or as part of a regimen in which, you know, a checkpoint inhibitor or another chemotherapy or immunotherapy can be given along with an injection, so that the immune system is kind of trained to respond to where the signal is, which in this case would be an injection in the peritoneal cavity. So there's a lot of intratumoral tumor injection protocols out in the world. Almost none of them are for patients with peritoneal disease. Probably none of them are actually, other than this one. So we really want to make sure that patients with peritoneal metastatic tumors have the same opportunities as other patients to be eligible for clinical trials, and to just kind of show a path that this disease is actually an opportunity to learn a lot about how these treatments work, and also just to say, hey, our patients really should be eligible for clinical trials as well, because we can do the same things, safely with experimental agents that are commonly done for patients with other cancers. And then lastly, and very briefly, I'll talk about our second clinical trial. This one's currently enrolling. Without bogging down in the details, there's one particular immune chemical or cytokine that is present at very, very high levels in the ascites fluid, the fluid in the peritoneal cavity, as well as in pleural fluid, or fluid around the lungs of patients with cancer. It's called IL-6. Or interleukin-6, and there is a FDA-approved medication that is commonly used for arthritis and other conditions that blocks that cytokine. And we have an open trial now using that drug. It's called tocilizumab, as an intraperitoneal or intrapleural injection directly into the body cavity to try to mediate or lessen the degree of inflammation, and hopefully change the immune environment in a way that could turn the immune cells back on, or wake the immune cells up, which is kind of a common theme. There's a ton of immune cells in peritoneal and pleural fluid in patients with cancers, but the cells are not doing what they're supposed to, which is kill cancers. So in future iterations, hopefully we'll be talking about the outcome of this clinical trial as really stimulating our ability to take cells from that fluid out into the lab, purify the cells, change the conditions, and reinfuse them back into patients in a TIL trial, or using the patient's own lymphocytes. You may have heard of TIL trials, not unlike CAR T cells, but a different concept. But just in layman's terms, to make those kinds of clinical trials available to patients with peritoneal or pleural metastatic cancer, who are so often excluded from clinical trials. We would like to show that not only should those patients be eligible for clinical trials, but they might be ideal candidates because we have access to the fluid and the tissues in order to learn how it's working in real time. So I think that's plenty for 15 minutes, and I will stop there and turn it back over to Dr. Turaga. Thank you.
Patrick, that was wonderful. I mean, I think I hope most of you on the symposium recognize the incredible work that's being done by Dr. Wagner, Dr. Bartlett, and their team at the Allegheny Health Cancer Network. And I think just exploring the idea of changing the microenvironment, understanding it. And I think the critical point that he made was around, the fact that patients are not often enrolled on clinical trials, but this may be such a unique opportunity. And so I think for any of you that are exploring clinical trials, this would be something where I would certainly say explore and consider. So thanks, Patrick, for that. I think our next speaker, Dr. White, may not be here yet. And so if it's okay, we had some pre-questions that I think was that were in our circulated piece. And so if it's okay with you guys, I thought it may be worthwhile for us to just chat about this some. I think the question for JP, if you're still here, and then maybe Patrick, you can also weigh in. The question around what about repurposing drugs? So maybe talk a little bit specifically. The question is around using ivermectin, mebendazole, febendazole, and aspirin for appendix cancer. What do you guys think about that?
I guess I'll go for it. So, I mean, I think the idea of repurposing drugs is a great idea, but they're not all created equal. You know, for example, the study we did with the atezolizumab and bevacizumab, you know, these are drugs that are approved in other indications and have been shown to work, you know, synergistically in like, say, you know, renal cell cancer, hepatocellular cancer. You know, very recent work from Andy Lowy and others at UCSD showing palbociclib looks to be active, which, you know, that's repurposed from breast cancer. You know, I would stay away from ivermectin, and you know, fortunately, I think that there's a lot of you know, maybe not good information about these drugs. I mean, it's you know, just you know, I think that in terms of how we repurpose, it's good to have some kind of idea of what you think that the drug is doing. This is a big part of why we're trying to get preclinical models so we can test the drugs in our, you know, cell line models and mouse models before, you know, moving into patients. That's actually a big part of why we felt confident to do our trial of paclitaxel, that it worked very well in the PDX models. So, you know, because there's not a lot of companies that are making drugs specifically for appendix cancer, repurposing, I think it's absolutely something that we have to do, but it's not, repurposing doesn't mean just take everything you can take on the shelf and try it.
Yeah. There's a question, and maybe Patrick, I don't know if you guys have explored this or if any of you have, but the question is, what are your thoughts on why appendix cancer patients get fewer liver mets than colon cancer patients? Have you guys seen any signals in your translational work? Or
That's
I
a
don't know.
really interesting question. And I mean, I think it's not just the liver, though. It's sort of liver, lungs. I think I mean, in my experience, it's, you know, it happens. It does happen sometimes. But I have often wondered that. I've often just thought that the appendix is so small and narrow calibered that, and this is a simplistic way of thinking about it, but it also doesn't, I don't think it has as rich of a lymphatic supply or a blood supply as the rest of the colon. So it's a little bit of an island, and it doesn't take long for a tumor to get through the other side of it. Those are more explanations about why it goes to the peritoneum, though, than why it doesn't go. But I wonder if the vasculature of the appendix has something to do with it. You know, the tip of the appendix does not have a rich blood supply, I don't think.
Kiran, if I recall correctly, I don't know if Ed's on, but I think the Wake Forest group looked at some of the genomics of the different primary tumors and metastases, sites of metastasis, and it looks like there's probably some molecular things that are directing where these metastases end up. He could speak to that probably better than I can, but that's been my understanding as well.
Yeah. Ed, are you still on? Because I think there's some burning questions on bromelain as well that maybe we should.
But I'll say no. The echo doctors. I mean, it's a super important question, because if we understood really, you know, what are the key requirements that allows the cell to metastasize, then that would give us a good target of how we could prevent that. You know, so if we really knew, you know, you need genes X, Y, and Z to allow the tumor to spread through the blood to get to the liver, that would give us, you know, things to target. But I think the difficulty is that it's complex, and that there's not just one way that the tumor can figure out how to survive in the bloodstream. It's multiple different ways, and so that makes it a little bit harder to understand. But I think, like Dr. Wagner said, I think the question is why doesn't it go to the liver, not to say why does it, you know, I think it goes to the peritoneum because it's just closer. You know, well, why doesn't it go to the liver? I don't know. Hopefully we'll have an answer for you soon.
Yeah. Andriana, another question that came up was again on the genetics of appendix cancer. The question was really just around, the person asking a question had a family member who had appendicitis and passed away. And, in hindsight, they're wondering, could this have been a genetic thing? So maybe both speak a little bit about what do we already know about genetic predispositions to appendix cancers? And then also, could you also talk about, should all patients with appendix cancers and appendix epithelial neoplasm, so Lamens, Hammonds, who should get genetically tested? What should happen there?
Sure. Thanks for the question. I think this is an important point. I think from the panel studies that we have, which are studies that look at known genes that increase risk of susceptibility to other cancers, we've seen about 1 in every 10 patients with appendix cancer present with kind of a change in their DNA that may increase risk for developing cancer overall. But we don't yet know if there are appendix cancer specific changes or susceptibility genes that may increase risk. GAP is actually going to be doing the spell check of the entire normal DNA called the whole genome to try and help get at that question and then look and see if those changes are reflected in the tumor to know that they're contributing to carcinogenesis. However, I think given that we see 1 in every 10 patients with appendix cancer, which includes all histological subtypes, I don't think we've yet had the data to be able to granularly look at Lammen-Hammond versus more goblet cell, signet ring cell, mucinous adeno cases there. I would recommend genetic testing for all patients diagnosed with an appendix cancer for multiple reasons. That proportion may also identify family members who may be at risk of hereditary cancer syndromes for which there are potential cancer prevention, surveillance, and early detection strategies. I'm not going to give away the data, but we're presenting at ASCO GI in the beginning of next year, looking at risk of second cancers among patients having been diagnosed with an appendix cancer. And some of that data would also be in support, I feel, of making sure to get genetic testing given potential risk of different secondary cancer types as well. So the answer is we don't know yet. We really need a comprehensive study, which is what we're looking to do to be able to glean one way or another. And I think both of those would either way, it's going to be an informative answer for the field.
Yeah, wonderful. Patrick, I'm going to ask this next question to you, which is, how close are we on a vaccine for patients with appendix cancer? Like what are you guys discovering from the immune microenvironment? And certainly I know there've been vaccine studies that you guys have done. So how close do you think we are to a vaccine?
Yeah, so vaccines are a very complicated, as you know, very complicated topic, and some vaccines are directed at it's kind of a one size fits all. Others are patient specific, and still others are actually cellular vaccines, which was a clinical trial that was done by Dr. Bartlett at when he was at UPMC a while back. I think it was a very small study, and I don't think we don't have active protocols in that area. I think I don't think we're that close. I guess is the answer to your question, but I don't think it's an appendix cancer specific question. I think how to make a tumor vaccine in a generic sense has not really been cracked yet for most, even for common cancers. So probably before there's a lot of momentum in rare cancers, we're going to want to see something like that take off in, a common cancer somewhere. Lessons learned along the way in common cancers help us in rare cancers. So I just don't know that we're all that close for a vaccine.
Yeah. Dr. Lambert, maybe if I can ask you a few questions and maybe you can help kind of answer a few questions in one. There's a few questions about, it seems like we're moving away from cytoreductive surgery and HIPEC for tumors. And, maybe can you speak a little bit broadly? And I know Dr. Levine covered this in the morning, but broadly, can you speak a little bit about chemotherapy, cytotoxic chemotherapy, and surgery? How do you kind of think about this for the different grades of PMP? So maybe just a general sort of overview of that.
Yeah, great. So again, I'm just going to come back to what we talked about at the tumor board, how critical the pathology is, right? Because that is where it all starts. And then from there is how I think about it. Like there's an algorithm depending on if this is a Lamb, a Hammond, or a low grade adenocarcinoma, and then whether or not there's peritoneal disease already present, or if it's a moderately differentiated or a higher grade, like a poorly differentiated signet ring cell or the goblet cell tumors. And then again, it really depends on how, when the person is diagnosed, is the tumor contained to the appendix? Has it already spread in the peritoneum? And then from there is what determines next steps in terms of whether or not somebody needs to have part of their right colon removed so that we can test the lymph nodes. As we talked about earlier, the Lamb and the Hammond, and even the low-grade adenocarcinomas have such a low risk of spreading to lymph nodes that we don't necessarily need to remove part of the right colon to test the lymph nodes, especially in the setting if the tumor has already spread in the peritoneum. But once you get into the higher grade tumors, the moderately differentiated signet ring cell, poorly differentiated goblet cell, now you have an increased risk of concern that there may be lymph nodes involved. So even if a person doesn't have peritoneal spread, they might need to have part of their right colon removed to really determine their stage. And then that's when we start thinking about the cytotoxic chemotherapies, and cytoreductive surgery and HIPEC may be a part of a multidisciplinary approach, or multimodal approach, I should say, to treating the cancer, to really try to effectively treat it as much as we can. But in those settings, we may be recommending cytotoxic chemotherapy first to test the tumor biology. I think we all would agree that if somebody starts on chemotherapy and the cancer just grows through it, they're not likely going to benefit from a cytoreduction in HIPEC. We really need to have some control of the disease before we put somebody through a big surgery like that. And also we can use the cytotoxic chemotherapies to help do some cytoreduction for us, and then optimize our chances of getting a complete cytoreduction at the time of surgery, and again, test that tumor biology. So that's how I think of them. I don't think of them as mutually exclusive. I definitely think of cytoreduction and HIPEC more up front for the low-grade tumors, systemic chemotherapy, cytotoxic chemotherapy for the higher grade tumors, and it's a combined multimodal approach. And figuring out the right timing for each person is dependent upon the circumstances of their tumor and their cancer.
And I guess, you know, maybe if I could just emphasize, so mainly it's the low-grade disease, it's generally surgical local regional therapies. I think for high-grade disease, certainly thinking about systemic therapies.
Yeah, and that's coming back to, I think, what Dr. Foote was explaining during the multidisciplinary tumor board about the rapid division of the cells that are more sensitive to chemotherapy, just how chemotherapy works in general. Obviously, we've got Dr. Shen, who's more of an expert in that than I am, but you know, that's the rapidly dividing cells that are going to be more sensitive to chemotherapy through intravenous chemotherapy. So that's why we tend to reserve it for the higher grade tumors.
Wonderful. I see Dr. White has joined. Hi, Mike. It's great to see you.
Yeah, thanks.
Mike, we're running a little bit tight on time, so I might request you to keep your presentation short if you can. I know it's hard to crush all that great work into a few minutes, but before everyone disappears, actually, there was one more group of questions. Maybe JP and Patrick, I can ask you guys, and maybe Mike, you can also answer this. Patients with high-grade disease who have pleural effusions or bone mets, how do you approach folks like that? And then maybe JP, if you could talk a little bit about how do you think about systemic therapies for someone like that? And Patrick, when would you do local regional treatments? Would you ever do high-talk? Would you ever do bone ablations and radiation, things like that?
Yeah, bone mets pretty rare, although I just had a recent patient with it. You know, for the most part, the bone is vascular, so the systemic therapy should kind of access the bone. So you know, if the disease is amenable, if it's the tumor is responsive to chemotherapy, that should still be responsive to the bone. But you know, if it's limited, hopefully that you would be able to do like a local regional therapy, like an ablation or similar.
Patrick, what about pleural effusions? Do you guys?
I think for us, you know, for most of us, speaking for surgeons, sort of generally, high-grade disease in the pleural cavity is generally not isolated, without there's usually peritoneal disease as well. And I think the risk of addressing pleural disease goes up. And I think it makes a bit more sense for us if we're talking about low grade disease where we feel like we can get a lot of mileage out of an operation, but sometimes the diminishing returns, with pleural disease. I think for bone mets, that's almost always going to be a deal breaker for surgery because unless it's an isolate, in a rare case where there's one bone met, I think maybe, we would still consider surgery, but generally speaking, we're not going to want to put patients through risk of an operation unless we feel like we can get all of the cancer out. And I think bone mets are, take us outside that box.
Great. Well, I think we have a few minutes. Dr. White's promised me he'll be able to get through our presentations quickly, and I think at 3:30 we break out for our regional sessions. But thank you all for answering all these questions, and those questions that you've had or have asked ahead of time that you want to be answered and we haven't covered those, feel free to put them in the chat, and we'll try to get to all of them as you guys are listening. So our last speaker today is Dr. White, who is an assistant professor at MD Anderson. I've had the pleasure of knowing for a long time. I think a superstar in the field, won the SSO award and the ACPMP award for his research. So Mike, take it off.
Great. So thanks everyone for being here. And before I begin, I just really want to thank the ACPMP for their really fantastic support in this work. This work that I'm going to show you today would not have been possible without the support of this group. And looks like I can't control. We can go to my first slide. Oh,
I think it's at your bottom left. You might see those little.
Yeah, I just got it. Thank you. So I won't belabor this point, but as we well know, the care of appendiceal adenocarcinoma is difficult and requires a lot of tools in our toolbox. Surgery being, I think, one of the most important, but obviously paired very importantly with systemic therapies and local perfusional therapies, and as we mentioned just recently, rarely radiation therapy. And one of the really exciting things in the last few years in oncologic care for patients has been the role of immunotherapy. And importantly, as we begin to utilize immunotherapy for a variety of GI solid malignancies, there are significant gaps in knowledge and understanding which patients will really benefit from immunotherapy. And in appendiceal cancer, that's especially important and is just beginning to be understood. So as we go into this, I think one of the important things and one of the things that we're looking for in this work is a biomarker. And what we define as a biomarker is a molecule or the presence of some type of cell type or protein that tells us whether or not these tumors would respond to immune checkpoint blockade or really any other type of treatment. But especially in this case, we want to pay attention to immune checkpoint blockade. Classic markers for immunotherapy are things like PD-L1 staining, tumor mutational burden, mismatch repair protein status, tumor infiltrating lymphocytes, and all of these are markers of an active immune system that's an anti-tumor immune system that's going to affect the ability of immunotherapy to have a strong anti-cancer effect. And importantly here as well, I want to highlight some work that Beth Helmick has done from MD Anderson as well in defining the role of tertiary lymphoid structures and the importance of B cells and not just T cells in driving response to immune checkpoint blockade. And we'll highlight that a bit later here today as well. But largely what we've been utilizing to date has been tumor mutational burden, PD-L1 staining status, and mismatch repair status of these tumors. And so this is work that Dr. Shen and I were happy to be a part of, working with the Natera company in profiling over 800 appendiceal adenocarcinomas. And perhaps frustratingly at first, we found that the tumor mutational burden was significantly lower in appendiceal adenocarcinoma than even rectal cancer, which has a relatively low rate of high TMB or MSI high status, and similarly, much lower than colorectal cancers, which a subset of those will respond to immune checkpoint blockade, classically if they're MSI high. But as you can see in the middle panel and the panel on screen right, the MSI high status and TMB status for appendiceal adenocarcinoma is significantly less likely to respond to immune checkpoint blockade by these more classic biomarkers. We were buoyed, though, by the observation that when we look at mutational signatures, appendiceal adenocarcinoma actually had a very large subset of patients that had MMR deficiency signatures in their whole exome sequencing, which was similar to the rates seen in colonic adenocarcinoma, which, as you recall, have a significant proportion of tumors that will respond to immune checkpoint blockade. Buoyed by this data, Dr. Shen and his group were able to perform really one of the first positive clinical trials in appendiceal adenocarcinoma, looking at anti-PD-1 atezolizumab and anti-VEGF bevacizumab, and were able to show that against the synthetic control arm, there was significantly improved survival in the blue line here as compared to the orange line. And this was especially important when we look at the time and duration on trial for these well and moderately differentiated patients, that you can see quite a few maintained on trial with some clinical benefit, while those treated with standard of care, which, as we know and have seen, have relatively low rates of response, had a lower or less time on trial and less clinical benefit than when compared to the immunotherapy patients. Importantly, however, we see that not all patients benefit from immunotherapy. There are some patients that progressed rather quickly, and this is where biomarkers come into it and play an important role in defining for us, hopefully pre-hoc, which patients will benefit and which patients won't benefit with immune checkpoint blockade. And when we looked at the clinical specimens from these trials and these biopsies, we hoped to perform whole transcriptome data, and came up with a very common problem that we approach with appendiceal adenocarcinomas, in that the mucin load in these tumors make them very technically difficult to sequence, because their tumor cells are rare, and all of these tumors failed qualitative or quality control analyses, and so we weren't able to garner any bulk data. We were, however, able to collect FFPE slides, and using a relatively novel technology, we were able to perform spatial transcriptomic analyses of these tumors. And what this spatial transcriptomic analysis does is it tells us the RNA transcription at each point on an XY axis for these slides, and defines what types of cells are at each point, and provides not only whole transcriptome data, but also the network that is occurring. So using an example from Marco Veroni's blog, we see the city here, and we want to not understand that there are schools and there are roads, but that the schools tend to be next to parks, and these different cells play different roles within the city and within these tumors. And even when we have a highly mucinous tumor like an appendiceal adenocarcinoma, when we put in a big patch of mucin, that doesn't take away from our information of the other cell types and the way that they integrate. And so we were really excited to run a pilot analysis, as supported by the ACPMP, of this CosMx assay to understand the spatial transcriptomic relationship of these tumors. And I'm excited to share the initial results of this with you today, that we found that in bulk analyses, we're able to differentiate tumoral tissue, normal epithelial tissue, as well as various immune infiltrates within these tumors. And then when we look at what these tissues look like under the microscope, we can then map these different cellular subtypes and their protein expression to XY coordinates, and we see tumoral tissue here on the left and immune aggregate tissues here on the right. And excitingly, as we look into these immune aggregate tissues, we began to notice that those patients that were alive with disease on therapy currently had what we, on neighborhood analysis, were these aggregates of B cells and CD4 positive T cells that were both interacting together. And these niches of the types of cells next to each other, which were called niche 2 in this setting, out of 15 different niches, were much more prevalent within the tumors of those patients having an ongoing response, and were much less prevalent when we looked at the tumors of those patients that had progressive disease while on therapy. And so as we unveil what is underneath these niche 2 niches, are these B cells, these CD4 positive T cells, and these interactions between the two. And you can see very well-defined architecture of these cells and their interaction in these patients that continue to have a response on immunotherapy. And when we look at our second patient that also continues to have an ongoing response, we see that niche 2 unveils a very well-defined germinal center of B cells within this tertiary lymphoid structure and CD4 positive T cells as well, and an interaction between these antigen-presenting cells and these cytotoxic T cells being activated against these tumor cells. When we see our patients, and I apologize if this is not projecting all that well, but the patients that continue to progress on disease, the rare times we do find these niche 2 cells, we see that the interaction of B cells presenting antigens to these CD4 positive T cells are much more disorganized. They aren't forming these well-defined germinal centers that we would hope to see. And this is true for both of our patients that unfortunately developed progressive disease, where you can see that the B cells in blue here are just sort of scattered throughout CD4 positive T cells and are not forming these well-defined clusters of cells. We see this sort of throughout our patients that unfortunately progressed on therapy. And when we look at bulk analyses, we find that CD4 positive T cells are not actually associated with response to disease, but that it's really the B cells and immune cells and plasma cells that are driving this response. And the antigen presenting cells really seem like they're the ones that are driving our response to therapy instead of bulk infiltrate. So obviously a lot of limitations to this work. We weren't able to identify bacteria, which we were really interested in. These oddly are very sterile tumors as compared to colorectal adenocarcinomas. But we really, in this pilot analysis, we're excited to find that we can get biologically plausible information from these specimens, and that it appears that we may have some early biomarkers of response to therapy that we're excited to look at in more specimens and to continue to validate them in a clinically meaningful way. So very quickly going through a lot of data, but exciting, and I really want to thank the group for their support in this work.
Thanks, Mike. That was wonderful. I have to say the explanation of spatial transcriptomics was probably one of the best that I've heard in a while. So that was fantastic.
Thank you.
I don't see any questions specific to Dr. White's presentation, but I do have a question for you, Patrick. I don't know if you're still here, but there's a question saying, is there any connection to IgA nephropathy to your new RIOT trial? Have you seen anything in that regard? Yeah, I'm not sure if Dr. Wagner is still here. Well, I think, it's about 3:32, so we're just a few minutes over. All of us are available. I think all these talks will be recorded and on the ACPMP website. And certainly, all the grant awardees would be happy to answer any questions if any of you have, and maybe direct them through Alyssa, through you, if they have any further questions for our grant awardees. So thank you all for your attention, and all the presenters, Mike, Dr. Wagner, Dr. Holovacci, and I guess that was it. Those are 3 of you. So thank you again, and it was great research, and looking forward to seeing all of you in our regional sessions.
All right. Thank you so much, Dr. Turaga, and all the speakers from part 3. And thank you again also, Dr. Levine, Lambert, all of our speakers, and everyone in attendance today. A special thank you to my fellow coworkers, especially Deborah Shelton, our executive director, and Margaret Najarian, who's helped me today as well.